As we come together today, I'd like to acknowledge the Wurundjeri people of the Kulin Nation and pay my respects to these traditional custodians of country throughout Australia and their connections to land, sea and community. I also extend my respect to all Aboriginal and Torres Strait Islander peoples here today. So it's a personal thrill to me to be introducing Eric Green today. Eric's in Australia to learn about the exciting developments that are happening in genomics here and to share some of his own insights, which will be invaluable. Many of you know Eric by reputation and will be familiar with his research. Since 2009, we actually worked out that the 15th anniversary is coming up of being director of the National Human Genome Research Institute in the US, which is one of the NIH institutes. The institute is one of the largest publicly funded genomic research agencies in the world, on an annual budget that we can only dream of. The institute is one of the largest publicly funded genomic research agencies in the world. Eric was the institute's scientific director for 7 years. He was chief of the NHGRI Genome Technology branch for 13 years and founding director of the NIH Intramural Sequencing Centre for 12 years. And as an interesting side note, I believe Eric is now Francis Collins' boss. For 2 decades, he's directed an independent research program that included integral start-to-finish roles in the Human Genome Project and groundbreaking work in mapping, sequencing, and characterising mammalian genomes. Eric, you're a hero to many of us, and I'm delighted to hand over to you. Welcome.
Well, thank you very much, Kathryn, for that very gracious introduction. You got it almost right. Francis, I'm not Francis Collins' boss. I'm this boss's boss's boss. Okay. I couldn't handle the responsibility of Kathryn reporting directly to me. But I am thrilled to be here. Thank you so much for your hospitality and so many of your staff who have just treated me just so well since getting here. And let me make sure we can advance this. Here we go. So what I am thrilled to be back in Australia. This is the second time I came here. And I do want to point out that the previous time I was here was almost precisely 12 years ago. And I came on for a similar reason. I wanted to know what was happening here then just broadly in genomics. This visit's even a little the same, but even more interested in what's happening in clinical applications of genomics. Last time I was mostly hosted by Simon Foote, who some of you may know, and he was then a dean of the medical school at a nearby university. And Simon and I worked together on the front line of the Human Genome Project. We were mappers. I was mapping chromosome 7. He was mapping the human Y chromosome. And we commiserated and worked hard and collaborated and got to be lifelong friends. It was during that visit that I also met this gentleman and have now struck up a lifelong friendship with Warwick, who then was the CEO of the NHGRI, and Warwick is right here in the second row visiting. And the important thing I want to point out about Warwick and I is 12 years later, we haven't aged one bit, not at all. We look exactly like we looked back then. So this was 12 years ago. I would have gotten here sooner, but a thing called the pandemic delayed things. And now Tiff and Daniel and others have been wonderful of allowing me to come here and have set up in a very informative, busy, but very gratifying week. And I'm on day 2 of 5 days of intense meetings. So what I want to do here, though, is really what I do, I'm going to sort of weave together my story of human genomics. And as you're going to hear as I tell the story, is that it is very much one that has a combined view of someone who has lived the entire story of human genomics, because I got involved in the field essentially at the beginning and have been involved for my entire professional career. But it's also very much through a lens of someone who has major responsibilities in funding human genomics research, at least in the United States. And what I have found is in telling the story of human genomics, it could be told in a book-like fashion. divided by 4 chapters, if you will, that follow an initial prologue. And the other thing I would tell you as I'm looking out at the audience is that what you're going to hear from this story is, for some of you, and depending upon your age, it's going to be a walk down memory lane. And you'll find that very entertaining because it's a walk down memory lane. For others of you, the trainees in the audience, it's going to be, some of it will be new, especially the early chapters. Maybe you wasn't quite familiar, but it sets a nice context for what you might be working on. So it will also be enjoyable. So whether you're younger or whether you're older, I think you're going to enjoy the story. I sometimes say that my talks are reminiscent of a Pixar movie. There's always something for everybody, whether you're a kid or you're an adult. And what I would say is that despite its relative youth, human genomics has thus far unfolded, I think, as a very rich and exciting story that can be told in a book-like framework. And so here's the table of contents. This is basically the outline of my talk. And I'm going to first introduce you to the prologue, and then I'm going to tell you about chapters 1 through 3. Those all represent the past, but we're really at a very exciting juncture, and that many of you, along with many of your colleagues around the world, are in the midst of creating the 4th chapter, which is about what's happening now and into the future. And I'm going to end, that's where I'm heading towards at the end of my talk. So I don't have time to give details about any of these chapters, but I can provide you some explanatory excerpts from each of these components. Well, let's start with the prologue. You know, the science of DNA grew substantially throughout the last century. You could point to various notable events. I always like to point to the discovery of DNA's double helical structure in the 1950s, followed by the elucidation of the genetic code in the 1960s as really pivotal in the metamorphosis of DNA sciences. However, the real prologue associated with the story of human genomics was the molecular biology revolution of the 1970s and the 1980s. And each of the components of this story will always have a big question, because that's what frames that part of the story. And the big question for the prologue was, can we develop the tools for comprehensively exploring genomes? Now, the molecular biology revolution brought us tools. They brought us tools for cloning and manipulating DNA on the far left, tools for reading out the letters of DNA on the middle, and tools for amplifying selective regions of DNA, such as those on the right. And these tools just kept getting better and better throughout the 1980s. And all of a sudden, rather than just using the tools to selectively look at this bit of DNA of an organism or this bit of DNA of an organism, we started to think about, was it possible that these tools could be utilized in a fashion that would allow us to look at all of the DNA of an organism, of course, all the DNA of an organism with its genome, and that got pretty serious consideration. And of course, there was particular interest in comprehensively studying the human genome due to the compelling challenges that we were facing in human and medical genetics that really required a better understanding of the structure and the function of the human genome. And so, in short, the advances in the tools of molecular biology and the growing excitement about the ability to start to think comprehensively about studying the DNA of an organism led to the coining of the word genomics and the naming of the new field of genomics, both of which happened in 1987. And in fact, some of you may not appreciate that if you go back and you look in the medical or the scientific literature prior to 1987, you will never see the word genomics once, because the word didn't exist. The word got coined in 1987 and first appeared in the medical and scientific literature in this opening editorial in a new journal called Genomics, where the editors laid out their vision of what all this was going to be about in this brand new field that had just been launched in 1987. Now, 1987 is a very significant personal year for me. I graduated with a combined MD PhD degree, ending my formal education in 1987, which meant never once during undergraduate medical school or graduate school did I once hear the word genomics, because the word didn't exist, which really says something about how you oftentimes have to become a lifelong learner, because I never learned about genomics because the word didn't exist, and everything that's happened in genomics for me has taken place after my formal education. But that's 37 years ago. If we think about why was a word invented and why was a field created in the late 1980s, it was simply because it was very clear that there was going to be an audacious new endeavor undertaken by the scientific community. And of course, that audacious new effort leads us to Chapter 1. That effort would eventually get to be known as the Human Genome Project, and it's reflected by Chapter 1, which covers the years of the Genome Project, 1990 until 2003. Now, the big question for the human genome in Chapter 1 was, can we actually map and sequence the human genome? Now, I want to pause here and immediately point out something about every one of these chapters, because each one of them that I'm going to describe was not written by a single author. It played out under the visions that were developed by multiple members of the scientific community, including the research funders and the scientists, that provided a blueprint for how you were going to pursue genomics during that particular time interval. And in fact, the Human Genome Project was guided by a series of these strategic visions, one after the other, that were instrumental in articulating and then re-articulating the goals and the plans for the project. Now, the Human Genome Project, by almost any account, was wildly successful. It accomplished all of its goals ahead of schedule, and it provided the world the first reference sequence of the roughly 3 billion letters that make up the human genome. I was, once again, extremely fortunate. And I had just graduated with my MD PhD a few years before. I was doing a clinically light residency in clinical pathology, which gave me an opportunity to be back in the lab essentially full time. About 2 years before the Genome Project started, I jumped into a genomics lab, which it just sort of wasn't even called the genomics lab at that time, but someone who was developing tools that were going to be used by the Human Genome Project. And then I found myself at Washington University at one of the first funded genome centers participating in the Human Genome Project, giving me a chance to basically, along with Simon, be on the front line of the Human Genome Project from day one and wrote it out to the end of the project. Now, the Human Genome Project, for those who have either remember it because you were living at the same time, or for those of you who read about it in history books, it was really unusual, especially for biomedical research. It was big science, which biology wasn't used to. It was consortium and highly managed. It was production and technology oriented. It had these highly focused goals. It had multiple funders from all around the world. It didn't involve generating and answering hypotheses. It was more about production. And on top of everything else, it was highly controversial. Oh, and by the way, it also wasn't guaranteed to be successful. And at times I can tell you it wasn't entirely clear that it was going to be successful, and that made it scary. But all that aside, at the end of the day, it was successful. It achieved all of its goals, including its signature goal of sequencing the human genome for the very first time. Now, this next slide will many times will show the dichotomy in the audience. For those of you who were getting Nature and Science in the mail, back in 2001, you will remember when these issues came in the mail, featuring the Human Genome Project's draft sequence of the human genome. Paper reported in Nature, and Science reported the efforts of a parallel effort by a private company called Australian Genomics. And so this was sort of the famous tie between the public effort and the private effort that resulted in these landmark publications that are existing actually in many history books and many biology textbooks. For those of you who weren't getting time and weren't getting Nature and Science back then, you may be getting them a few years ago, because those same two journals featured the fact that 20 years earlier they had commemorated, or they had published those major papers, and there was that look back at having the draft human genome sequence 20 years earlier. And then last year, of course, we and many others around the world celebrated the fact that we reached the 20th anniversary of the completion of the Human Genome Project, which ended in 2003. So last year, 2023, we had lots of commemorative events and various ways of reminiscing about what had happened. Well, every chapter has highlights. So what were the highlights of Chapter 1? Well, without question, the top highlight was that we did it. We actually generated the very first sequence of the human genome, and that will go down in the history books as an incredible achievement. I always want to point out a couple secondary highlights that I think are worth mentioning. First thing was a really important legacy of the Human Genome Project was this ethos of data sharing. And for some people, they may say, well, everybody shares data now. That wasn't the case back then. And the Genome Project really broke that open by saying, we will share data. We will share it even before we publish it. We will share it even before it's perfect. We will share data much more aggressively than ever had been shared before. And it created an ethos that carries forward. We're still working at this. There's lots to do at an international level. At any given country, there's lots of issues, but at least it's part of the conversation now. And I think the Genome Project deserves credit for pushing that agenda. The other thing we did different was from the very beginning of the Genome Project, integrated bioethics issues into the project, sometimes referred to as the ethical, legal, and social implications, or ELSI research. And that has been a part of our efforts from the very beginning of the Genome Project. And even today, my institute, we dedicate 5% of all of our research dollars go to studying the ELSI research program. In other words, studying the bioethical aspects of what we are doing. And so this, therefore, I would say were the main highlights of chapter 1. But as is always the case, you end one chapter, you start another one. And as soon as the Genome Project ended 21 years ago, we immediately began thinking about what's next. And what's next related to chapter 2, which really saw the field grow beyond the highly focused goals of the Human Genome Project, bringing a major expansion and maturation of genomics. And it's really important to appreciate that the different funders around the world that were involved in the Human Genome Project, including the 2 major funders in the United States, they came together for different reasons, such that when the Genome Project ended 21 years ago, it should not be very surprising that the different funders went off and did different things in genomics because of their core reason for getting involved in the first place. And in fact, what happened starting 21 years ago was a massive dissemination of genomics all across the life sciences. And shown here are just 9 representative areas where genomics has had a major impact over the last 21 years. Each and every one of these are deserving of a lecture, a symposium, or even entire journals and fields. I'm not going to talk about any of this because the area that we moved to at the National Human Genome Research Institute, part of the U.S. National Institutes of Health, of course, was thinking about applications of genomics to health and disease and to medicine. And it's important for you to realize that, in fact, it was very important that we were able to think about what were we going to do beyond the Human Genome Project at our institute, because we were actually created, we didn't exist until the Genome Project. The US Congress created us to lead the US's effort at the NIH in the Human Genome Project. And obviously when the Genome Project ended, we needed to redefine why we needed to exist. And so in fact, that was what we were thinking about in chapter 2. And the big question we thought we needed to help lead was, can we use genomics to advance knowledge about health and disease? And so we saw the importance of having a well-crafted vision that would articulate what should go on in genomics related to human health beyond the Human Genome Project. And so what we did was not dissimilar to what went on during the Genome Project. We engaged the scientific community, we strategized with them. We did it for the last 2 years of the Genome Project, so that literally the day the Human Genome Project ended, in April of 2003, we published this paper in Nature, which was our strategic vision for the future of genomics following the Human Genome Project. This vision represented a major pivot for the Institute, and I would contend the field, because we were pivoting from the Genome Project to what was going to be next in terms of health applications. And every one of our strategic visions always have a sort of an iconic figure that you could point to. And you can see here the iconic figure which summarized the vision at the time was the pivot was all about building now the structure upon the foundation of the Human Genome Project, that bedrock that provided the ability to now connect genomics to biology and to health and to society. And in doing so, see a major expansion on how genomics was going to be used across the biomedical landscape. And so this strategic vision guided us until 2011 and really represents the key aspect of chapter 2. Now, what was the most sort of fundamental aspect about this next phase of genomics following what I refer to as the pivot? Well, we had to go back to basics. The reason NIH went to the table of the Human Genome Project, and the reason we could convince the US Congress to give money for us to help make a major investment in the Human Genome Project, was the following. It was that we recognized that so much of what we do in medicine was about managing the average patient, And in fact, I can tell you everything I learned in medical school was about how to treat the average patient. But we knew we could do better. We know that each of us is unique. We have uniqueness related to our physical and social environment, but we also have uniqueness with respect to differences in our genome. But we had no access to that genomic information. We didn't know much about the genome. But if indeed we could get to the point of having information in particular about the uniqueness of each patient's genome, maybe then we could actually use genomics to improve the practice of medicine. The idea of the pivot was, how do we get there now? It should therefore not come as any surprise that whether you looked at the popular press where the Genome Project ended, such as New York Times Magazine, or the scientific press like Science, they put the two words together. It was the most obvious thing to do, genomics and medicine, and you came up with the phrase genomic medicine. And that was clearly where we needed to be heading. The problem was back in 2003, 21 years ago, we didn't really understand what genomic medicine was about. We could sort of spell it, but the concept was blurry. And we needed to bring genomic medicine into focus. Now, we could define it, and in fact, we were able to sort of think about what this might look like. So it would be an emerging medical discipline. We'd use information about each patient's unique genome and use that somehow to tailor their medical care. But the fact of the matter is that made sense. But in fact, of course, we had to figure out what that was going to really mean and how we were going to get there. Now, I will point out that there are related but not quite identical terms that have been used interchangeably along the way: personalized medicine, individualized medicine, more recently, precision medicine. You will hear me use the phrase genomic medicine because it just talks about genomic information, which I think is a more narrow subset of what gets talked about compared to these other phrases. These are all important phrases and are all relevant. I just tend to stay more narrow since I'm a genomics person, just focusing on genomic medicine. Well, how were we going to better understand what genomic medicine was going to look like? How were we going to get there? Well, it did remind me of what things were like when the Genome Project began, that we had a starting point and we had a journey. This was just a different journey. And I was also a different starting line. Because here now the starting line was the Human Genome Project's completion. And the finish line was going to be see genomic medicine realized in some fashion. And it seemed an almost insurmountable journey at first, but then you just got to break it down. And you say, okay, we're just going to take this one step at a time. And we have to understand we don't even know what all the steps are. And there are going to be twists and turns, and it'll be scary, and we'll have to face major hurdles, but we can get there. And the other thing you do about this is that you say, every single time you've gotten somewhere, you should take credit for it, and you should sort of check it off the list and feel gratified and just keep going and moving closer and closer to realizing your ultimate goal. We also appreciated that this was going to take far longer than the Human Genome Project. And just like the Genome Project, it wasn't going to be a sprint, and it wasn't going to be one person. It wasn't going to be one discipline. It wasn't going to be one country, not one funder. Like the Genome Project, but even more so, this was going to be a marathon. And there are going to be many of us from many countries, many disciplines, really running shoulder to shoulder for a long time. And we had to be prepared for that. But just focus on one step at a time. So what was the first step? Genome Project ended 21 years ago. What was the number one thing we had to deal with then? Well, it was pretty obvious. We laid out a lot of things that needed to be done, but the very first thing was that we had just sequenced the human genome for the very first time. So we had one representation of the human genome, and we knew ultimately we wanted many human genome sequences. Ultimately, we wanted genome sequences of our patients. The only problem is that we were incredibly celebratory in the fact that we completed the Genome Project. We were celebratory that we had sequenced the human genome for the first time, but we also had to face the realization that while this was incredible and it was a remarkable achievement, it was also expensive, because that first sequence, the human genome, cost something on the order of $1 billion, roughly, which was a bargain for everything it has accomplished. But we appreciated the fact that where we needed to be, especially we're going to thinking about sequencing genomes in a diagnostic way, was we needed to knock a lot of those zeros off. And in fact, in the strategic vision that we published the day the Human Genome Project ended, we actually gave the figure of where our goal should be to be able to develop technologies that would allow us to sequence a human genome for $1,000. And in fact, this became a battle cry for the community, that we needed the $1,000 genome. And the problem is, once again, we had no idea how we were going to do that. We knew that what had gotten us to the billion-dollar genome, where large factories with expensive equipment and people working 6 years of active sequencing for the human genome and hundreds of people working 7/24, that's good for a billion-dollar genome. We need a $1,000 genome. And in order to do that, we had to develop something all brand new, almost science fiction-like, because that's what was going to be the $1,000 genome. And we knew we were going to need all new technologies. As an institute, we created a technology development program that put out funding opportunities for all sorts of different ideas for sequencing DNA. And we absolutely saw some remarkable things happen, both successes and failures, because we entertained, we encouraged high risk. But we get only partial credit, because a lot of credit also belongs to the private sector. It attracted a lot of venture money. Companies came in. I think it was a brilliant, beautiful example of public-private collaborations, such that we were able to see, and far faster than any of us possibly could have imagined, a reduction in the cost of sequencing. And in fact, the rest is history and has been chronicled and graphed by us and written about, because we've done it. We've gotten past the $1,000 genome threshold. In fact, we have our eye on the $100 genome now. So we did that, which is remarkable. By the way, I vividly remember that up until the pandemic, at this point in my talk, I would always say, I would argue, and I would, in my very biased way, I'd say I would contend that NHGRI's technology development program that yielded the $1,000 genome was the #1 best example of NIH doing technology development that had a high impact. Then the pandemic came along, and we actually had NIH heavily involved in developing COVID vaccines. So now I always say the $1,000 genome, it's in the top 2 of the 2 most impressive technology efforts ever undertaken by NIH. And that's okay. That's good company to be in. So we're in the top 2, and we're happy to be #2, but it's still quite incredible. And of course, what that means is we can check that off our list, and now we go to step 2. Well, step 2 was all about understanding how we vary, because it's all very clear that we were thrilled by having a sequence of the human genome, but now we really wanted to know what the differences among people, and then we want to start to be able to catalog those differences, and we knew that's where we had to be heading by having a much better understanding about the human variation. Now, we've known for years the different kinds of variation. I'm not going to give a big lecture. Many of you, I'm sure, know the different ways the genome can vary from single nucleotides to various other types of changes. But, we didn't know a whole lot about what a typical person's genome looked like, nor did we have very much information about where across the 3 billion letters do we typically have variants in which populations and so on and so forth. Numbers have been thrown around, but it is roughly thought that at the time the Human Genome Project ended 21 years ago, we only knew about 10,000 places in the human genome, for example, there was a single nucleotide difference, only 10,000. And we knew there was hugely larger numbers than that. Well, what did we do as the cost of sequencing went down? Well, we used those sequencing methods, and we generated hundreds of genome sequences, then thousands, and then tens of thousands, hundreds of thousands, and now millions. And with that, we've gotten more and more information across different human populations, with far more studies needed to even get more diversity across different populations. But we've also begun to understand more realistically, what does a typical person's genome look like? When the Genome Project ended, there was always assessment about differ about 1 out of 1,000. We now know from now sequencing millions of genomes, we could be much more refined in that estimate. And we could know that on average, a typical person's about 99.6% aligned the same with a reference genome. And with the variation in terms that comes, you can just sort of see across the different types what that really looks like. Those numbers aren't as important as just appreciating that we now have better understanding when we sequence a person's genome, what we might expect in terms of diversity. And the bottom line is each of us is incredibly similar. Roughly 99.6% of the nucleotides are completely the same across people, but it's that last 0.4%, of course, that's different, that has a lot of rich biological information. To understand that rich biological consequences of genomic variation, we must understand how the human genome actually works. That has to be step number 3 after the Genome Project. And it is important to remind people that the Genome Project never promised anything other than an ordering of the letters. We knew it was going to be Herculean efforts to elucidate the functional coding of information across the 3 billion letters. And we also knew that was going to likely take multiple generations of scientists to completely tease out all the functional landscape of the human genome. But just think about where we are now, where we're compared to where we were 21 years ago. We've been staring at those 3 billion letters. Major efforts have come and continued. Some have been retired in code efforts, many other efforts around the world. And many individual investigators all around the world have fed in lots of functional information about the human genome. And all this is collected in databases and data resources. And this is the way we view the human genome now. And I'm sure many of you spend time looking at browsers like this, which I always like to explain, especially to non-scientists, is basically the Google map of the human genome. It allows you to zoom in and zoom out. We're not looking for hotels and gas stations. We're looking for genes. We're looking for transcription factor binding sites. We're looking for conserved regions. We're looking where it gets made RNA, et cetera, et cetera, et cetera. We have got an incredibly detailed understanding of a small fraction of the functional elements across the human genome. We should be incredibly proud, but we should also be realistic. There is far more that we don't know than what we do know, but especially I'm sure there are many of you in this audience who are helping us push this iceberg up so that more and more of it will be above the layer of the water. But we should recognize that there's a lot of incredible complicated information encoded in those letters. And over time, and with many efforts that are continuing, we will basically go from just knowing a tip of the iceberg of what is currently known to really having a much more complete understanding about things like the function of every one of our 20,000 genes, all the complicated epigenomic language and marks across our DNA. Thinking a lot about, which is getting a lot of attention, I know, in multiple places in the world, the appreciation for that other nucleic acid, RNA, and methods for being able to not only sequence RNA directly, but to be able to detect modifications of RNA and being a very important molecule in the choreography of how genomes function. Better and better understanding of the complex transcriptional regulation of these 20,000 genes. And of course, there's lots of other omics to be thinking about, proteomics being among them, but there are others as well. And so we have to be realistic and saying, yes, we should be proud of where we are in understanding how the human genome works. But while we want to continue to infuse great young talent into helping us tease through all the complexities, because I am quite convinced my grandchildren will still be interpreting the human genome sequence and maybe even beyond that as well. So what are the highlights of this first attempt at expanding and maturing the field of genomics? Well, I think we have to declare the new technologies for sequencing DNA. By the way, as I got walked around yesterday in various institutions just within 8-10 blocks of here, I must have seen 30 or 40 sequencing instruments, right? So I mean, this is just so routine now. It just wouldn't have been the case 21 years ago, seeing that diversity of sequencing instrumentation and just seeing it utilized by in so many different ways, in so many creative ways around here and really around the world. Top highlight. I think we should appreciate how far we've come, where we went from once knowing maybe 10,000 places in the human genome where we vary. We now know of hundreds of millions of places in the human genome that we know that we vary, and it's getting better and better all the time with more knowledge coming out about human genomic variation. And we've made a lot of progress in identifying the functional elements of the human genome, with a lot more that still needs to be done. So, we could take a step back and we could watch how we're doing on this journey. We could do an instant replay, if you will. When we knew this was going to be our journey, and we first, we reduced the cost of sequencing a human genome more than 1 million-fold. That deserves a pretty nice looking check mark, right? We then went on and we sequenced millions of human genomes. We learned a lot about the genomic variants, and we've made resources available where we can be understanding and studying and characterizing those variants. That deserves a check mark. We've advanced profoundly our understanding about how the human genome works. Maybe it doesn't deserve a full size, but it deserves a nice looking check mark. And then even, when it's set therefore up a circumstance of we're about 14 years ago, we're about 2010, it was very clear that we had begun to see major steps in that journey to genomic medicine be realized. So much so that it really began to be clear to many of us, including me, that it was time to even push harder to think about clinical applications of genomics. And chapter 3 was very much about beginning to apply the tools and the resources of genomics and thinking about putting us en route to genomic medicine implementation. And I think the big question right around 2010 or so was, can we actually demonstrate effective uses of genomics in medicine? Now, this is, again, a significant point in history for me, because I had literally just taken the baton in 2009 from Francis Collins, who was the previous director of the Institute. And he had kicked off a strategic planning process, recognizing we needed a new strategic vision for the new decade in 2010 and beyond. And I had to complete it after I became director. And I once again relied on lots of brilliant people, including some sitting in this room, to help shape what that vision was going to look like. And I pushed very hard for this idea that we needed to really embrace the idea that genomic medicine was within striking distance, that we should be pursuing that. And so in 2011, when we published the strategic vision, the first under my directorship of the Institute, we actually put the phrase genomic medicine in the title, which actually once again created some controversy, which I've overcome. I think it's great that people were concerned and we proved them wrong. And the iconic figure really described and predicted what the progression was going to look like, and going from a fundamental understanding of the genome to actually improving how we can apply healthcare using genomics, with some predictions about when those achievements were going to be achieved over different intervals. And it was very clear that the key aspect of going beyond those first few steps in that journey was to now really start to hone in on understanding genomic variation as it pertains to its influence on genome function. All of you appreciate the fact that not most variants actually are inconsequential. There's only some of them that actually influence genome function, and only a subset of those actually have any substantial impact on our traits and our phenotypes, and as people interested in human health, those particularly related to human health and disease. The problem is we don't know that relationship. We just know that in general, only a subset. And the little variants in the genome don't hold up their hand and say, I'm relevant and I'm not. You got to figure that out. And an oversimplification, but let me just remind you, is we do believe that the vast majority, any one of us contains something like 3 to 5 million single letter differences in our genome compared to the person sitting next to you. That's just one of many numbers you can cite. The vast majority of them, if those 3 to 5 million are blue, a vast majority of them are completely inconsequential. Only a small subset shown in orange have any biological relevance, and even a smaller subset shown in red are actually medically relevant. Well, we want to know that. We want to know that for as many variants as we can figure out, which ones can we just ignore because they're biologically inconsequential, and at the other end, which are the ones that we really should be paying attention to because they're medically relevant. And so this becomes a key priority for understanding the relevance of genomic variation in health and disease. In terms of describing progress in this area, it is useful to think about what has happened in the last 21 years in particular by thinking about the continuum of genetic diseases that relate to the different contributions to disease risk. And here there is this a dichotomy that's often used between common and rare diseases, which is an imperfect it's an imperfect dichotomy. It's really more of a continuum, but it does tell the story nicely when you explain what has happened in terms of genomic impact. So on the one hand, there are rare diseases, something like 8,000 of them exist that are known to exist so far. But rare diseases are usually genetically more simple, because usually it involves mutations that appear in one gene that is the major risk factor for getting that disease. And yes, there might be other variants that influence severity, and yes, there might be some environmental influences, but by and large, you're looking for one gene. And because it's more straightforward, we've made more progress. And I would just like to point out, and we've seen the acceleration of this over the last 15 years in particular, that's just breathtaking. When possible, I like to cite numbers. Rare diseases, which many of you know, cystic fibrosis, sickle cell, Huntington's disease, and so forth. If you go back in time when the gun went off on the Human Genome Project, October 1 of 1990, there were 61 rare diseases that we knew what the mutated gene was, 61. One of them was cystic fibrosis, and one of them was sickle cell. So that was known before the genome, 61. Sometime in the next month or two, we're going to go north of 6,000. So we've gone from just 61 and nearly now we'll go over 6,000. That is just incredible. And of course, it has led to incredible numbers of insights about fundamental biology. It's also led to diagnostic and therapeutic opportunities, and we could have story after story after story of triumph. It is amazing what has happened, and genomics deserves an immense amount of credit. Rare diseases are really important, and obviously they're devastating to families and patients. However, in aggregate, it's not what fills this hospital. It's not what fill the hospitals up and down the street or the clinics. What fill hospitals and clinics in this city and everywhere else in the world are common diseases. Hypertension, diabetes, Alzheimer's, dementia, cardiovascular disease, mental health issues, cancer, et cetera, et cetera. These are the common diseases that are incredible healthcare burdens worldwide, but they turn out to be complicated because, of course, it's not a single gene that have variants, but rather it's multiple variants with what is typically a much greater collective contribution of the physical and social environment. This is hard. But we've come a long way through a variety of major areas of study involving statistical associations between variants and that allow you to make correlations between getting risk for a disease like diabetes or hypertension and a particular region of the genome. In a few cases, we've been able to go in and actually figure out what the gene is or what the variant's doing biologically, but for the most part, we're a long way from that. However, we went from having very much of a framework for studying common diseases to now having nearly 500,000 or 400,000 associations between very discrete regions of the human genome and a very important health-related trait or a disease. And now we just got to get across the finish line on these, and this happened. It hasn't quite happened as much as we wanted, but we'll just keep plugging away. And it's lots of optimism of why we will see lots of progress over the next 10 years. I do want to point out that this is a continuum, and of course, there's a lot of complexity in between, and many diseases sort of have elements of being common and also being rare, and I acknowledge all that, and it's all part of the complexity of human biology. So I think we should marvel at this progress. We should particularly be proud of rare diseases, and we should be optimistic about that we're setting things up to be quite impactful, and what we'll hopefully discover about more common diseases. So if we go back to what we've been watching, I think we can say there have been significant advances in unraveling the genomic basis of human disease, although, again, mostly for rare, less so for common. So, we'll give us a medium check mark. Well, this, therefore, sort of we've seen this progression with respect to getting us all the way through studying human disease. But I haven't said anything yet about medicine, because studying diseases is not practicing medicine. However, I think we can say that we've begun to see, starting just a few years before the pandemic, and certainly now, it's here. We finally begun to see vivid examples of genome medicine now emerging. Now, I will stress, it is a small check mark. It is just the earliest examples. But what has transpired is that by the middle of last decade, things really started to get exciting. We were applying all the tools that we needed. We had cheap genome sequencing, we had catalogs of genomic variants, and we also were getting better and better functional understanding of the human genome. And wow, we were starting to identify especially rare diseases practically on a weekly basis. And these developments all came together with some of the earliest examples of genomic medicine being implemented. Now, I want to stress, while these initial examples are just the tip of the iceberg, what they really provided us, as much as anything else, was clarity. We finally could say, we now know what it is like to actually use genomic information as part of medicine for the first time. And that has been incredibly influential and incredibly helpful. So let me just tell you what some of these examples are, because frequently what I'd be asked, okay, so what's on my hit list for what I would like to say are some of the earliest examples of genomic medicine being implemented. But before I give you the actual examples, I should point out that I have, as someone who's passionately interested in this slide in particular, because it was really what I was pushing so hard to see, unlike the previous parts of my story where there was a lot of logic and there was a lot of, sort of things were very sequential and predictable, things weren't as predictable here. There was not a grand overarching plan for how genomic medicine was going to be implemented, but rather what I've seen play out are different clinical niches emerged, each with an acute diagnostic need or a technological capacity. And there was a circumstance, in some cases, I could say in the United States, because maybe an insurance company was willing to pay for it, and they just sort of organically grew up, and they're very different, and they're all very illustrative. And there are 5 of them. So what are the 5? And I don't have time to describe these in any detail, but I'll just give you just 2 or 3 sentences about each. I think far and away what will be the grandest of impact in the long run in medicine will be cancer genomics. Cancer, as you know, is a disease of the genome. We already have seen immense amount of tumors whose genomes have been sequenced using the same methods you would sequence normal cells, and have learned a lot about the derangements that now are more commonplace in certain types of cancer. Some of that has been operationalized for diagnostic improvements. Some of it has been operationalized for therapeutic improvements. And I believe in the long run, we're just going to see the complete sea change in how oncology is practiced because of genomics. And so I think there's an immense amount of excitement there. Second major area where genomic medicine is here and now is, and even in this building, is in rare genetic disease diagnostics. I am amazed that in my lifetime, I now have seen a full progression where patients come in the door and you don't know what's wrong with them, and now you just sequence their genomes and you figure this out. And it's getting better and better all the time. And there's various clinical settings. In some cases, it's urgent in the case of a setting of acutely ill newborn. And the idea that you can sequence a genome in a day or two and save a baby's life, it happens all the time. And it's remarkable. And I can't believe now it is here, we are in 2024, I could say genomics saves lives. And that just wasn't the case 5 or 10 years ago. That's just truly amazing. The number one genomic test in the world, again, talk about an opportunistic niche that has come out, has been in the setting of prenatal testing, where the traditional methods of more invasive methods like amniocentesis and chorionic villus sampling have for decades been used to access fetal DNA for for parents who are interested in getting information about aneuploidy in their unborn child. But now with our fancy new methods for sequencing DNA, which are exquisitely sensitive, allow us to detect the small amounts of fetal DNA that are naturally shed from the placenta. The placenta DNA represents fetal DNA, and you can do as a screening method through a simple blood draw an assessment of whether or not there is aneuploidy present in that unborn child. It is the number one genomic test. It is estimated that between 7 and 9 million pregnant individuals will get a non-invasive prenatal test done this year. I know it's taking place in this country. In fact, I think it's taking place in this building, if I remember the tour yesterday. Amazing, amazing that this happened. It's becoming mainstream around the world, which is remarkable. Pharmacogenomic testing is certainly something that's been talked about quite aggressively. Why is it that people respond so differently to medications? Well, some of it is because there's differences in our genomes that are influencing drug metabolism pathways, and we're increasingly learning about that. And we increasingly are learning that a lot of this can be very relevant for commonly prescribed medications. I think around the world, it's pretty consistent that the uptake has been slower than any of us anticipated. There's lots of reasons for it. I know it varies from country to country. I do believe we will see a greater uptake. There's lots of barriers that need to be overcome, but I am quite confident that pharmacogenomics is going to become much more utilized in the coming 10 years as we learn more and as we improve our ability to implement this and get healthcare professionals to use pharmacogenomic information. The last area I'm going to just unpack a little bit, because there's some things I think it's worth emphasizing of what's here and now and what's not quite here and now in the arena of prevention, because this other stuff is a lot of this is about medicine, but now I want to talk about prevention, because I think there are genomics-based prevention that are absolutely here and now, in particular in the case of rare diseases. And you can think about this in various settings, and I know this is taking place here. Whether you have this gentleman who might be from family history, more common to a clustering of colon cancer, maybe the family gets screened. Yes, they have Lynch syndrome variants. And sure enough, that saves lives, because by screening that individual, that family, you can get more frequent surveillance. And if they get colon cancer, it'll be caught at an earlier stage. That's absolutely genomic-based prevention. Similarly, either at a newborn period or at some period, maybe we would learn that this little girl or maybe her family would have mutations in a gene that with a simple diet adjustment will live a normal, healthy life. If you don't impose a diet change, this child could have developmental delay and very significant consequences. But through prevention, either at a newborn screening situation or maybe in another setting, you can save lives. And this is absolutely here and now. These are rare diseases, a lot of excitement around what's going on, and common diseases. And of course, the rage here are polygenic risk scores. Can we look at people's collective set of variants, stratify them, and make predictions about whether they're at high risk or not at high risk for something like early onset diabetes or dementia or early cardiovascular events. I think this is incredibly exciting. My institute is funding a significant amount of work in the implementation of polygenic risk scores, but it is not something I think is ready for implementation in the clinic quite yet. Maybe I'll take this icon away within a couple of years and we see what some of these studies say, but I would contend this is still a research area, not an implementation area. So what are the highlights associated with seeing this en route to genomic medicine? I think it was that 2 slides ago. It was those compelling examples of genomic medicine having emerged. We really are here. We have finally seen genomic medicine, and it is now a much clearer concept. Some of this has come about because of progress in rare disease understanding, and some of it, I think we should also celebrate the fact that we have in place a nice framework for studying more common diseases. And so we really have seen this journey accomplished, albeit with the last step just being the earliest. And so it really was a time to think about this current decade that we're living in. So as we approach 2020 and the start of the new decade, it was apparent that progress in human genomics continued to be breathtaking and celebratory. We realized the strategic vision that had guided us 10 years earlier was, and 10 years in genomic time is a long time, it needed to be updated. And it was really time to have a new strategic vision for the new decade. And the real question for this new chapter was, can we deploy genomics more broadly, maybe even equitably in medicine? And that very much is about what chapter 4 was about. We started strategic planning before the pandemic. We ended up having to finish it in the middle of the pandemic, and we published this in 2020. And what is really important for me to emphasize is that this strategic vision was very different than any of the previous ones we had put out. And there's lots of ways I could describe it. I'd like this quote that actually came out last year, but it really it encompassed what we were already thinking back in 2018. A non-scientific magazine, The Economist, which was talking about the Human Genome Project, wrote this quote, and I think it says everything, that no part of biology is untouched by the consequences of the Genome Project. Genomics has therefore come to form a framework for biology in a way that the periodic table forms one for chemistry. It touches everything. So we went to strategically plan starting in 2018, and even when we published it in 2020, we could no longer strategically plan for all of genomics because genomics was touching everything. And rather, our role in leading this effort had to be much different than what it had been for any of the other times where we had put out a strategic vision, because genomics now was being done everywhere. Every NIH institute was doing it. It was just fundamental. We actually rebranded ourselves. We have a new identity that we used as part of the strategic planning process, where we now say we're not about all of genomics, we're about the forefront of genomics. And that was used as the framework for the strategic vision that we published in 2020, and we even put the phrase of the forefront of genomics in the title of the paper. I would encourage you to read this. It's 10 pages in nature. If you've not read it, please do. If you want to read it again, please do. I'm not going to describe it in detail. I'm just going to tell you about the 4 categories of things that we described. Because one of the most important things we learned in the strategic planning process, and it really comes across in the paper, is understanding what it means to be at the forefront of genomics is all about what we need to be doing in providing leadership for the field. And what we finally realized was that being at the forefront of genomics is all about being responsible stewards of the field. And that meant to sort of take responsibility for 4 domains. The first domain didn't even involve money, didn't even involve experiments. It involved integrity. That at the forefront of genomics as responsible stewards, we should be leading efforts to make sure we have the guiding principles and values for the field, and that we should be articulating, re-articulating, extending them. And we described 9 of them, and it relates to things like diversity and equity and social justice, open science, data sharing, data standards, all these things that we've been fundamental. We were told at the forefront of genomics, we need to make sure these things remain at the forefront so that everybody doing genomics abides by these fundamental principles, everything from diversity to data standards. We also heard that somebody, meaning us, at the forefront of genomics needed to deal with the foundation on which everybody uses genomics. If everybody's doing genomics, they all rely on data resources. They all rely on software tools. They all rely on materials for improving genomic literacy and thinking about the tools of genomic data science. We had to sustain that foundation. We needed to improve it. And that was our responsibility at the forefront of genomics. A third element at the forefront of genomics is recognizing what's impeding progress. 21 years ago, it was the $1,000 genome. What is it today? And if we could break down those barriers, everybody benefits because that barrier no longer exists. And we described 6 of these things in the paper, such as DNA synthesis, DNA editing, characterizing genomic variants at scale to figure out which are the blue ones, which are the orange ones, which are the red ones, thinking about how to actually implement genomic medicine, because implementation is really hard in healthcare, and we need to think about what are fundamental barriers that exist and how can we knock them down. And the fourth area when you're at the forefront is be audacious and do compelling big research projects. And we give a handful of about half dozen examples, thinking about what does every human gene does. You know, we only know a small fraction. What is the fundamental underlying architecture of common diseases? What is a comprehensive view of all the regulatory elements in the human genome? These big audacious projects, and we're pursuing those as well as others. And it's these things all together, working together, that what it means to be at the forefront of genomics. So the last thing I want to stress is that I keep saying genomic medicine, genomic medicine, genomic medicine. Sometimes people think, I don't think there's a lot of things upstream anymore that are important. That's not true. I just emphasize the destination because it's very exciting. But trust me, I know that we need to continually, for the next decade, make better and better reference sequences of the human genome, especially those for more diverse populations. We need better and better technologies related to, let's get down to a $10 genome sequence. Let's think about all the RNA sequencing we want to do. We'll be doing that for the next 10 years. Of course, characterizing genomic variants will take us many more than even 10 years. Understanding how the genome works, 10, 20, 30 years. And we, of course, have many more things we need to accomplish in understanding human diseases, not to mention needing many more examples of genomic medicine being implemented. And we don't just make slides like this, we actually come through on projects. And if anybody ever looked, if you want to, this is just eye candy. This is a subset of the consortium that NHGRI supports. And you could see, you can bin them into very basic research as a priority, more translational research, clinical research, and bioethics research. We're covering the whole territory. What I want to leave you with is the very last part of the strategic vision, which was something that we had never done before in our strategic visions, where in this latest 2020 strategic vision, we ended it by making some predictions. I think some of it was the pandemic, that we were so bored because we were home and we'd written the whole paper by Zoom. We said, let's just have some fun. Let's say 10 crazy things and put it in print. And so we strategized as an institute, and we came up with 10 bold predictions of what might theoretically be possible by 2030. And they were audacious and they were high risk, but they were fun. And so we ended it near the very end of the paper in this box, and that's the 10 that are listed there. And of course, then Nature said, well, you have to say something about them because you can't have a display item unless you refer to it in the text. And so, okay, so we said here in the text, we'll say something about box 5. And so he said, but now more than ever, solutions to these problems seem to be within striking distance. Towards that end, and with the characteristic spirit of genomics audacity, we offer 10 bold predictions of what might be realized in human genomics by 2030. And that's all we said. And then the press saw this, and that's all they wanted to talk about, were our 10 bold predictions. So having realized that, I turned to our communications people. I said, we undersold our bold predictions. People think they're a lot of fun. We need to bring our predictions to life. And so this is how my communications group brought our 10 bold predictions to life.
Genomics, which is the study of all DNA in an organism called the genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward from the success of the Human Genome Project, a massive reduction in the cost of sequencing a human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future, a future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully attained by 2030, achieving even one or more of these by that time would have lasting impact for science, medicine, and society. Prediction 1, sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2, the role of every gene in the human genome will be known. Prediction 3, environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4, genomics will no longer use social constructs such as race in human research studies. Prediction 5, student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6, genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7, it will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8, a person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9, advances from human genomics will benefit all of society and not just a few. Prediction 10, genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020SV. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
So I realize that I'm having way too much fun up here. That's why I went a little over telling you my story of human genomics and the excitement associated with the past, and particularly the excitement that we're going to see as we all create Chapter 4 together. With that, I'll end, other than to say we love to stay in touch with everybody around the world. I have a monthly newsletter. If you want to get emailed to you, and I'm very active with my team on social media, 3 different platforms. I'll leave this up for those interested. And with that, I will thank you for your attention. I look forward to the conversation we're going to have now. I'm on.
I'm on.
There you go.
I'm on. So, Eric, thank you for that tour de force. It's just wonderful to reflect where you come in at different times during that history into the genomics journey. And I was also thinking, and I must note here at the Murdoch Children's Research Institute, we were established as the Murdoch Institute for Birth Defects in 1986, which is a year before the term genomics was actually coined. So our journey as an institute has very much followed the journey that you've talked about. So we've about 300 people online. We'd love to hear questions from the audience. And I actually there are floating mics, I believe.
Yep.
Yep.
Right,
So right behind you to Richard Safri.
Thank you so much, Eric. It's a real privilege to have witnessed that talk. And as someone who remembers those heady days of the 80s and 90s, it was exciting to relive it, even if it's just a little bit. So many questions, but I'll ask one you've probably heard 100 times, and that's this ongoing lack of correlation between the heritability of phenotypes, which are very easy to see and trace, and the lack of a correlation in the genetic risk associated with them. And those two things continue to come up as an issue. Are you in the Peter Vischer camp that says heritability is all in the genetics? We just don't know where to look, or are you more in the It's the gene environment and other interactions that explain that gap.
You could, being a government person and probably trying to be politically savvy, I wouldn't take either extreme. First of all, I am certain there's going to be significant physical and social environment contributions, but I can't help but think about my iceberg graphic and believe that as long as there's so many important things below the surface of the water, we shouldn't dare think we're in a position to really even answer your question. I firmly do believe even some of the areas I articulated will play some fundamental role in accounting for that the difference of heritability or understanding of heritability.
Question up the back. If everyone could sort of put their hands way up and I can direct people to you with a mic.
Thank you, Eric. That was a wonderful talk. As someone who began her PhD in February 2001, I really enjoyed the summary. Eric, as researchers, as research institutes, some of the priorities around breaking down barriers to implementing genomics in medicine, they're not always things we can control or things that are a part of our day-to-day research. I'd love to hear your thoughts on priorities for how researchers can form the relationships, have the influence to break down some of those barriers that perhaps might rest in healthcare organisations and not under the control of research.
Yeah, no, it's a great question. There's a talk I give on Thursday in Sydney, which we deliberately are making it a different talk. I have a new slide I've never used before, which I don't know. I show a group of a family where they have a kids table, having dinner where all the kids are at the kids table, and then and make some joke about going to the adult table, and that's exactly what I feel like. I feel like for so long. Especially when we were working on the Human Genome Project, we were a bunch of kids at the kid table. For so long, it was just figuring out how to map and sequence, interpret the human genome. And then literally the second we touched healthcare, it forced us to the adult table. We have to talk about mortgages and insurance and interest rates and politics, all the things we never had to deal with at the kid table. So I know exactly what you're talking about because I am amazed at what I've even seen here at NHGRI, National Human Genome Research Institute, you would think we were well poised to deal with this onslaught of issues associated with when you try to change something in healthcare. And we weren't, because until we started to see any sort of healthcare, all of a sudden you realize, oh my gosh, this is so many complicated issues. So I know exactly what you're talking about. Your question was, how do you deal with it? Part of this is, and I think you see it in the way we form some of our consortium, is the recognition that when as you're doing the research, you need to have these interdisciplinary teams, which include people who are just as good at running a nanopore instrument as they are in understanding public policy, because the fact of the matter is the second you start to now have the ability to deliver genomic information at scale, blah, 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 you're going to need to think about how you're going to, at least in the United States, fight with insurance companies or deal with a regulatory agency, or how you're going to educate physicians, or how you're going to improve get more genetic counselors or improve the education of nurses and so forth. So it's like you're at the adult table. You got to act like an adult. You got to sort of build up an organization and support consortium that will make sure that happens. So I have felt it as I've had to staff up, even at the Institute over the last 10 years, in ways we just weren't needed before.
And Eric, it made me think of a comment you made around the Human Genome Project and that ethos of data sharing. And
Yes.
I think also for me, a foundation of the ethos of collaboration and
Yes.
international collaboration. How do you think the attitudes now are from that broad data sharing, given cybersecurity and, when you think about barriers in genomics, it's how do we navigate around that?
Yeah, it depends what day of the week you ask me what keeps me up at night. But we are, as you know, there is, data sharing was so important and so fundamental. And now there is, I know it's going on in more countries than just the United States, there is some concern about broad data sharing, about especially human genome sequence with all countries, some concerns around some countries, and the things that may be imposed would be absolute barriers that would make the international community of data sharing just fall apart. And so we're we're dealing with this, and some of it is not fact driven or data driven. It's based on concerns that I don't think have much scientific undergirding, but that doesn't mean they don't get pushed. And so this is a challenge. This is a challenge. And, for a long time, we thought it was people's concerns that were going to hinder us. I actually think people and patients, they want to see their data shared. I think these are political forces. And so this is a problem.
Great, we can talk about that offline.
Yeah.
We've got a question from the
from online. Yes, we've got
online.
many questions online. Very vibrant selection. So Jessica asks, what do you think, Eric, are the unique or meaningful ways in which Australia can contribute to global progress in genomics medicine?
Yeah, well, so it's a great question. And the short answer is it's part of the reason I'm here. I mean, you have this opportunity. These are all experiments. None of us know exactly what's going to work out well. And we all need to experiment. And in the case of genomic medicine, everybody, unfortunately, or fortunate or unfortunate, we're being forced to have a bunch of different flowers bloom because every country has a different healthcare system. And so inherently, you will see different experiments being done. And what I would say is be aggressive, be bold, and let us know how you're doing. That's why I'm here. I want to know how you're doing. And then I'm going to try to figure out, because I do believe that progress in one country might really help catalyze our ability to, at the adult table, win the argument that need to be changed to have progress take place in a different healthcare context, because every country will be different. And what I would say is, please share your data, fight like stink to share the data, be as participatory as possible in every one of these global efforts, of which the numbers just keep growing because there's so many different dimensions. But have people at the table of all these efforts of sharing information and best practices about genomic medicine implementation, about cohorts, about data repositories, about standards with GA4. There's so much great stuff going on here. Please share, and be at the table.
And I think another element to that is the reach into Southeast Asia, and
that's right.
That's
you'll bring others along that we have trouble bringing. That's right.
Yeah.
No, that's absolutely
right. Tiff?
Yes. So MJ asked a question about your, in discussion about the public-private partnership associated with Human Genome Project. What lessons have been learned there, and how important is ongoing engagement with industry in the context of progressing genomic medicine, but also implementing genomic medicine?
Yeah. So if the question was really the public-private partnership during the Human Genome Project, I'm not sure that was that wasn't a partnership. That was a little bit more.
It does say divide.
Yeah. So that was, but that said, I think, especially since then, there are many great examples. I mean, there's many great examples, and I certainly think sequencing technologies is among them. I think we constantly look for opportunities. Sometimes they're good partnerships, sometimes they're not as good partnerships. I frequently get asked about, and I understand here there is a market for not direct-to-consumer genomic testing. I frequently get asked about that, I think that there's many examples of very good productive interactions, public-private, and many examples where that we get a little more concerned that they're a little more aggressive than we think the data should show. But keep an open mind and so on and so forth. I just think whenever possible, I've been thrilled to have companies, especially diagnostic companies, who are willing to share information about variants. When we could win them over, everybody wins, right? So I just think, as always, we want to see keep open mind, open communications, and see when there's opportunities for having these partnerships, because I do think there's a lot of individuals in the private sector who see how everybody benefits when we work together.
We'll take one more
All right, one more question, and apologies to those online that we didn't get to your question. Mark asks, among your 10 bold predictions, my favourite is the elimination of the variant of uncertain significance by 2030. What, in your view, are the most likely breakthrough approaches that will achieve the same?
Oh, wow. There's like some real experts sitting right behind you that you could probably ask those questions of. So I would say, I would say it is such a high priority, and it's such a hard problem that it is attracting around the world some of the best and the brightest. In some cases, not even pure geneticists. I mean, in some cases, computer scientists, experts in artificial intelligence. It is gratifying to see what we're able to recruit. And so I think maybe the answer is going to be, it's not going to be one modality, it's not going to be one program. I think it's going to be the fact that it is so important and so hard, but so interesting. It is attracting a multidisciplinary groups of people coming together. And I actually also think that there's a spirit of working together in efforts like ClinGen and others that is also very international in flavor. And so I guess I get very optimistic by the people power that are being brought to this and the intellectual human capital. It's the human capital that it makes me optimistic.
And a final question from
Sure.
me. How do you continue to keep your Congress excited about genomics?
Oh, wow. You know, so here's what I'd say is, first of all, we have great stories. I mean, we got great stories, and they are compelling, and it's you know, when you can tell them the progression of what's happening, it does capture their imagination, and you know, I'm biased, as I'm sure you are too. You know, the moonshot was incredible, and everybody marveled at the moonshot. You know, it's been incredible what the moonshot brought us, but in some ways, it's almost easier to show how this. You know, you don't have to point to too many people's lives who you change, or you really, and that resonates. And so, you got to keep it simple, and you got to make it compelling, and it has to be story-like. And so I find that easy, and actually they're fairly receptive. And for the most part, except around the edges, I mean, we've got a lot of massive political problems in the United States, but for the most part, genomics is not, you know, it's not about vaccinations, and it's not about, you know, embryonic cells, or, you know, so I think people in general on both sides of the aisle like genomics and like what we're doing. You got to dance a little bit around how it all gets paid for, and et cetera, but I think by and large, the stories tell themselves because they're so compelling.
And I must say, you're a very compelling storyteller, Eric. It's been wonderful to have you here. And I would agree that ethos of sharing that you talked about, the collaboration that has underpinned everything about the Human Genome Project, and you talk about the value of human capital, that all is exciting. And I certainly think that's what excites us here, both our international partnerships that are very vibrant. I think genomics has really brought that to life for so many of us, and the great people that want to keep solving the problems. So thank you, and we look forward to continuing to work with your wonderful institute.
Likewise, thank you.